business. This is the first episode in our 4-part webinar series. Today, our topic is chromatographic techniques for the characterization of CQAs in biosimilars. Our speaker today is Andrew Argo. Andrew is the strategic marketing lead for biopharma and new modalities in consumables and lab automation. He's held positions across industry with a focus on analytical method development for LC and LCMS applications for products ranging from peptides to gene therapy for almost 10 years. He is currently the strategic marketing lead for the biopharma and new modality portfolios within the chemistry organization, helping to understand and map out trends within these wide-ranging spaces to facilitate the prioritization and development of novel applications and products. Just some housekeeping items that we like to go over before the webinar begins. Your microphone and camera are switched off by default. If you have any questions, please feel free to submit a question through the Q&A chat box. We'll be happy to take the questions live at the end of the webinar.
Okay,
and with that, Andrew, go away.
Thank you, Leslie. Again, my name is Andrew Argo, the strategic marketing lead within the Waters organization. And today we'll be talking about chromatographic techniques for the characterization of CQAs, specifically within biosimilars. Very high-level agenda. Today we'll cover just a few specific techniques, charge variant analysis, glycan analysis, and peptide mapping, and how these actually apply to biosimilar characterization. So when we look at a biologic, specifically in this image, a monoclonal antibody, what you'll see is it's made up of a variety of variants that are native to the actual structure. And when developing a biosimilar, not only do you need to match just the basic sequence and structure of the actual monoclonal antibody or whatever recombinant protein that you're developing, but also should be matching to the greatest extent the variants that are present within this biologic. It's very critical that they are as similar as possible to make sure that the efficacy is similar when compared to the originator. A few different techniques, again, that are required to be leveraged to actually characterize these critical quality attributes, again, include charge variant analysis, glycans, and peptide mapping, all which provide their own individual types of data that can be leveraged to show, again, the biosimilarity between this product and the originator. So we're going to start with charge variant analysis. So we look at the wide range of products that are available through the Waters organization, specifically the consumables organization. You'll see that we do have, again, just a very large number of offerings, specifically with the charge variant, with charge variants products. You'll see that historically Waters does not have maybe as wide of offering within the ion exchange chromatography space, but this is beginning to change as we see the importance of charge variant analysis, not just providing information related to acidic and basic variants and so on, but also being able to dive in deep and actually identify and characterize the actual identity of these charge variants. And we're leveraging this and doing this by quite a bit of development on our end. Specifically with the initial release of the BioResolve strong cation exchange column, which will be the focus on this particular section. So when we look at charge variant profiling, there are two techniques that really kind of dominate this space. Again, ion exchange and capillary electrophoresis, which come with their own specific pros and cons and kind of operate within specific spaces here. When we look at ion exchange, it's characterized more by a lot more reliable and robust separations, can handle a lot larger sample loads, which allows for a variety of different secondary techniques, purification, to move on to different other types of characterization techniques if need be. It actually is also characterized by much greater run-to-run reproducibility. While capillary electrophoresis it is typically characterized by much higher resolution fast separations. Fortunately, it has been historically disconnected from secondary assays, though that is beginning to change in industry. And one of the bigger pitfalls is more of the small sample loads that it can handle, which unfortunately minimizes a lot of the ability to isolate and run those samples in secondary assays for further characterization. So when we look at cation exchange of Mab-based therapeutics, ion exchange can actually determine and monitor a variety of charged species through different heterogeneities through oxidation, asparagine deamidation, isomerization, and so on. And many of these CQAs are very critical, again, to the actual product themselves, and should be monitored and characterized to the greatest extent possible. Each one of these attributes can actually affect the efficacy of the product as well as the immunogenicity. So it's very critical, again, to not only characterize to the greatest extent the originator, but also the biosimilar product as well, and ensuring that they match as great as possible. So looking at the basics of ion exchange chromatography, it's a relatively simplistic idea, and really it's a particle, be it an anion exchanger or a cation exchanger, with some charge either on the surface or for a non-porous particle, or even if it's porous throughout the entire particle itself. But this charge is opposite to the actual analyte that you're attempting to actually separate. So an anion exchanger, you'll see, have a positive charge and is able to actually interact with negatively charged species on its surface, and vice versa for a cation exchanger. So this is a very basic principle, again, for the actual separations. It's much more complex than this, but again, all you're doing is trying to interact with different charges to allow for the separation of these particular charge variants. So when we look at the development of these of an ion exchanger, it's critical to understand the actual particles themselves, the actual chemistries that are being leveraged, and that's going to be important for the method development and actually the ability to develop specific methodologies. When we look at, and one of the ideas that we need to kind of understand is this concept between strong and weak ion exchangers. These don't particularly mean anything in terms of how tightly the protein is bound, but actually are correlating to the presence of the charge and over the pH range. So when we look at a strong ion exchanger, those are typically going to always be charged, while a weak ion exchanger, and you'll see specifically weak cation and anion exchangers, will be charged over a smaller pH range. And this is going to limit some of the actual method development that can be performed, but can also be a strength based off of how you actually develop your method. Specifically with weak ion exchangers, you can actually start to leverage different salt and pH gradients at the same time to allow for some very unique separations. And beyond the actual characteristics of the particle itself, you also need to understand specific characteristics within the protein as well. Ion exchange, again, is going to actually separate based off of, at a high level, the actual PI of the protein itself. But a lot of this is also majority is driven by the actual surface charge of the protein as well. So though the PI will kind of get you in this at the initial kind of information to understand exactly where and how you should be developing your method, it's very critical to understand that this technique is actually going to separate based off of surface charge. And again, when we look at PI, what we're actually specifically talking about is the pH at which the net charge on the protein is 0. And then above that and below that, you'll have your negative and positive charges. And what you want to understand is and characterize are the actual amino acids present and how they contribute to either a positive or negative charge. So all this information is going to be critical to understand how whether to choose an anion exchanger or a cation exchanger, and then how you should develop your methodology in terms of pH and salts as well. And you'll see on the right there some typical amino acids that are present, natural amino acids that contribute to the actual overall charge of the protein. And again, understanding how those are going to contribute, especially again in terms of being on the surface or if they're buried within the protein themselves, and how that's going to affect your separations. So when we look at the separations, how this is actually, again, the basic idea. The charged species is going to interact with your ion exchanger. And what you'll see is then you can leverage either pH or salt gradients. And specifically here, you'll see a shift and change in actual salt content. So attempting to bind at a low ionic strength mobile phase to begin with, and then increasing that salt concentration or increasing that ionic strength over time to outcompete the protein on the ion exchanger to then allow for that separations. And again, it's as that ionic strength increases, you'll then begin to see the elution of your proteins in specific charge variants off of the ion exchanger. And yeah. So again, the basic principles that we discussed in this from in the beginning is the idea that, of course, we're simply separating based off of the ability of the protein itself to interact with the ion exchanger, and then modifying mobile phases through specific ionic strength or even pH. But what we want to discuss is some of the basic ideas behind actual method development for an ion exchange separation. So the concept is very simple. We're actually having some charge-charge interactions, and then eluting base off of either modifying pH or ionic strength. There are a lot of different ways to go about performing this method development. Starting with the actual selectivity, optimizing with pH. So when we look at a variety of different chromatographs here from pH 6 down to 7.2, you'll actually see this is going to be a very critical and important piece of altering selectivity within your ion exchange methodologies. So you see, obviously, as pH changes, and we showed earlier on the high level view of how pH shifts charge based off of the PI of the protein. You'll see that obviously shifting throughout different pHs, you're going to be changing the surface charge that's available for the separations. And you'll see moving in this specific example through ribonuclease A, cytochrome C, and A protein. You'll see from pH 6 to 7.2, we're actually able to, again, alter the selectivity enough to where initially the first three peaks or a few of the peaks are actually co-eluting, and we're not able to actually see some of the charge variants that are present in the sample. But then moving to pH 6.2, 6.4, and 7.2, we actually start seeing the peaks not only spread apart and offer better separations, but we also begin to see swapping of different peak positions. So this is a very important, very important piece of the actual method development. pH is one of the most critical pieces to understand in terms of how it's going to affect your method development. Again, and this is why very important to understand the actual PI of your protein and characterize that as much as possible before you begin the process of method development. We can also, again, optimize retention by adjusting ionic strength. So again, when we look at a few different proteins here, we have five in this mixture. You'll see on the right, you'll again see that we're initially loading with very, very low ionic strength. In these particular examples, we're loading at 0 sodium chloride, 0 molar sodium chloride, and increasing up to either 0.15, 0.3, or 0.5. And you see that obviously with much higher ionic strength, specifically the 0.5, we get significantly lower retention and less separation of these species. And you'll see that even a couple don't actually potentially bind and actually come off right in the void during the injection. And then as we actually lower the actual ionic strength, we get much better separation, but even peak broadening here. So it's a balance between, again, separation abilities, and then again, making sure that you get sufficient peak shape for things such as limited detection and quantitation. Changing buffer may also improve selectivity. When we actually look at what buffers that we can leverage here, again, we looked at sodium phosphate as well as MES buffers here. And these do buffer over a similar pH range, but their overall the overall range where they can actually buffer is differs significantly between phosphate and MES. And what you'll see is the maybe the specific pKa's and the characteristics of these individual buffers will actually significantly alter not only again the actual separations between all three peaks, either increasing or decreasing, but you also start to see peak swapping that can occur based off of differences within the actual buffers themselves. And not only the actual buffers, but then the actual counter ions that are leveraged within these buffers can alter the selectivity to a minimal extent. So you'll see the differences between sodium chloride and potassium chloride. We may start to see as we move to potassium as opposed to sodium, you may start to see some differences in the ability to further separate and tighten up specific peaks. Now, again, this is a minimal effect, and some of these come from solvophobic effects. How their preference to actually interact with the ion exchanger as opposed to staying in the mobile phase itself. So there's some complex characteristics of these counter ions that can actually affect your separations. And again, if you're performing some very complex separations of a variety of different proteins, it may be worthwhile to look into how these different salts can actually affect your separations. And then one of the last pieces we'll touch on is temperature, of course. So looking at temperature, and historically, obviously, when you the idea that a lot of individuals may have is moving towards something like much higher temperature to allow for faster methods. Unfortunately, what you may start to see is when we look at 45 degrees Celsius at the very top, is that though you may start to pull apart peaks more, you may actually begin to see broadening of these peaks as well. So this may not be an ideal temperature to work with, though it may allow for faster separations. You may want to move down to lower, again, 40C, 35C, 30, and then potentially even cooling your column to alter the selectivity enough to, again, allow for sufficient separation of some specific charge variants, while also maintaining the peak shape required for, again, quality limited detections that may be critical for the development of a biosimilar. And you'll see these as we move down to lower temperatures, the peak shape begins to significantly improve. Though we do lose that separation between these peaks. Again, it's a balance between all of these characteristics. And as we move, as the industry moves not only to proteins but also to different modalities such as nucleic acids, et cetera, the actual characteristics and the way that these will begin to change, you'll have to alter temperature a lot more, again, as these structural characteristics are significantly different between proteins in those particular modalities. So looking at ion exchange, again, critical points here. It's separations based off of surface charge. When we think about different techniques such as ion exchange and capillary electrophoresis, again, the capillary electrophoresis specifically is separating solely based off of that PI, where actually denaturing is solely based off of PI, while ion exchange itself allows for separations based off of the actual surface charge. So it's much more dependent on the structure of the protein itself, which for biosimilarity is going to be very critical. So choosing that, though you may get sharper peaks within capillary electrophoresis, the actual structural characterization through chromatographic ion exchange may provide much more information, critical information to determine biosimilarity. Again, anion exchangers, cation exchangers separate based off of the biomolecule charge at a defined pH, and that pH is going to be one of the most powerful characteristics leveraged to alter selectivity. And then method development, again, beyond pH, can be modified based off of the ionic strength, pH, as well as combining a variety of those characteristics together in combination with either your weak or your strong cation exchanger. Okay, so we're going to move forward to glycan analysis. Again, Waters offers a wide variety of technologies within the glycan characterization space. You'll see that we have everything from columns to a number of other consumables to allow for, again, the very high level order characterization from the tertiary structures down to the individual glycans themselves, even to the point if we actually break those down into individual sugars, we can actually characterize those as well. So we operate across a wide variety of spaces within the actual Hillock space for glycan characterization. The reason why we do this is because there's no single technique that provides all the information needed, again, to determine not only just to characterize, but also determine the biosimilarity of our protein products that we're looking at. And this is going to be critical looking at the intact structure itself for a glycoprotein, performing characterization at that level, down to different proteases and digestions to allow for much more discrete separations and determination of glycan structures, down to, again, breaking these down through a variety of enzymatic processes and even hydrolysis to characterize monosaccharides. So it's very important to have every bit of this information. Not one or any other type of analysis is more important than the other. All pieces of this are critical, again, to determine the structure and characteristics of your biosimilar. So looking at glycans and why specifically we leverage Hillock separations. These glycans are very hydrophilic and again do not do well within standard reverse phase separations. Based off of the moieties that they're bound to, you may be able to provide, and we'll discuss that later, the separations through reverse phase, but the information you get may not be, you may not get the depth of information you need because it's not the ideal separations based off of the hydrophilic nature of these moieties. So again, they're highly polar compounds, which makes them ideal for Hillock separations, though they don't have a native chromophore. So it does make the actual detection difficult and requires either mass spectrometry or some different chemistry to allow for the modification and the addition of an actual chromophore. They can also contain a variety of acidic groups as well, the sialic acids, which makes them amenable to some different mixed mode separations where we can introduce an ion exchanger into this and allow for some different selectivity to allow for further characterization. And with that, though, these acidic groups do become much more of a challenge. And what we have to think about is the design not only of the particle themselves, but the actual column as well, and how that's going to interact with these very acidic species, as they can cause issues with recovery and even kind of that discrete separation between these highly complex sialylated glycans. So some general principles of hydrophilic interaction chromatography or Hillock. Ideally, this separation is going to happen between a water layer and the biomolecule itself. Now, that's an ideal separation. We understand that based off of the design and the ligand leverage within the actual column itself, you may get some different interactions, you may get hydrogen bonding, some different ionic interactions as well. But again, the ideal separation is just between the water layer that exists within the particle and the actual molecule itself. The gradient separations happen from high organic to low organic, again, opposite of reverse phase. And this is very important to understand, as this will change the diluent that can be used. Obviously, at this point in time, water is going to be your strong solvent. So having something in these particular species in a lot of water and injecting high amounts of water can actually disturb your separations and cause problems. And so so we have to take that in consideration, especially the solubility at high organic with these particular species. So it's something to take in consideration. So though it is a powerful technique, there are some caveats. And this has historically been used to separate a variety of small polar compounds. So we can take some learnings from that as well. So when we look at glycoprotein characterization, looking at method complexity and the actual depth of information you can get, we kind of have a variety of different spaces we can look at, specifically the intact protein, more of the middle up or minimal digestion approach, depending on if we're talking about MABs or just general recombinant proteins. We have the glycopeptide mapping approach, as well as the release glycan analysis approach. We won't touch on the monosaccharide analysis, but that is a space that we can and do offer solutions in if that is of interest. So starting with the intact protein, this is again one of the more kind of quote unquote simplistic Hillock separations in terms of the data that you'll actually receive. So what we're doing with this is simply taking the protein in its native form. We're not doing any sort of digestion, and we're trying to understand the level of glycosylation that may be present. So are we seeing two spots of glycosylation? We're seeing one, or we're seeing zero. And this is very important to understand, again, because we may not get discrete identifications of particular glycans, but this will provide us an understanding of how well the cell is actually not only producing the protein, but actually modifying it through glycosylation. And then you'll see here the ability to actually separate these species. But the reason why we're not getting, again, that discrete separation of individual glycan types is because the protein itself is driving a significant amount of the separations here. So not only are we leveraging the hydrophilic nature of the glycans, but we're also leveraging the hydrophilic nature of the protein too, which minimizes how much separation we can get. But if we want to gain some more information, we can, and when focusing on proteins like MABs, we can actually leverage more of what we call a middle up approach, which is provided through kind of this minimal digestion with some specific enzymes, and in this case, IDS digestion, which cleaves at the hinge region of the MAB itself and releases two different again specific pieces of the protein. And then what we can do from there is either analyze that as is, the Fab2 and the FC as well, or we can denature that and release those light chains from the variable regions of the heavy chain, and then analyze the light chain itself, the FD, as well as the FC region. This is going to allow us some more in-depth kind of discrete separations that initially, when you look at something like reverse phase, we'll just simply get 3 different peaks. So though this does provide us some more information than a standard intact mass analysis, unfortunately, we're still not getting the information we need in terms of, again, those discrete glycoforms that may exist. But if we, again, move to helix separation, what you'll begin to see is some specific separations based off of the particular glycans that are present. So if we think back to the intact mass analysis, initially getting just a handful of peaks based off of the actual presence of a glycan, what we're seeing now is not just the presence, but the actual specific types of glycans start to pop up. And this is due to the fact that we're chopping up the protein, we're minimizing the reliance on the separations by the protein itself, and the glycans are now beginning to drive more and more of the separations. So we're able to see more, again, discrete peaks that are related to particular glycans, giving us the ability to identify these either through mass spec or then begin to track them through just a standard UV-based chromatographic assay. Again, as we move through this process and we start to gain and want to gain more information, we can even leverage kind of a glycopeptide mapping approach. And we look at glycopeptide mapping. Anyone who's done peptide mapping, essentially what we're doing is, as opposed to performing the digestion itself and then leveraging an enzyme like PNGSF to release the glycans, what we're actually doing is keeping those glycans onto the actual individual peptides themselves. Now, this may not seem to provide a whole lot of information, but what we're actually able to do with this is confirm not only the presence of glycans, but that we're confirming the exact spot of glycosylation through assays like mass spectrometry, where we can actually fully characterize those peptides. And again, this is going to be critical for determining biosimilarity. Again, not only just through the presence of, but actual finalizing where that glycan is bound. But when we look at techniques like reverse phase that are typically used for peptide mapping, if we look where the green bracket is showing, unfortunately, we start to compress a lot of those, a lot of the glycans, the glycosylated peptides, because the peptides themselves are driving majority of the separations in a reverse phase technique. But if we were to leverage HILIC, this is where we actually begin to see much more discrete separations, even more than the middle up approach. And this is, again, due to the glycans driving majority of the separation. So we're starting to see significant separations and retention of these glycopeptides that allow for much better detection capabilities, as well as the actual glycosylation pattern determination as well. So again, that's as we take the protein and kind of minimize it further and further through a variety of digestion techniques, those glycans will start to drive most of the separations, and we'll get a better understanding. And we zoom in here. This is what you would typically see on the top in the red box. We zoomed in further from the initial chromatogram. Again, majority of the peaks are compressed in on themselves. And again, this is just due to the peptide itself actually driving the separations. And then the bottom, again, leveraging a similar gradient slope, we're able to further pull apart and see some very discrete separations based off of the actual glycan type itself. What's very important with this, again, is even at this level, we can begin to characterize through mass spectrometry these very low abundant species that may not have been visible through things like a middle up approach. We're not getting as much information. So this allows us to dig even further. And then one of the final techniques, and this is where we can potentially argue what is more important, what's more, which one gives us much more information, either glycopeptide mapping or release glycan analysis. They are potentially kind of, in my eyes, a little bit more equal in terms of the information they're providing, as the glycopeptide can provide actual sites of glycosylation, while release glycan analysis removes that actual site specificity. So they're kind of going toe-to-toe in terms of the information and the depth of information. It's just they provide different types. But for release glycan analysis, there has been a variety of conventions leveraged, specifically reductive amination. And this is what it's very laborious. It's a tried and true method, but it takes a long time, and it is minimal in terms of its ability to be leveraged on mass spectrometry or other different assays. And what we wanted to look at was how can we actually take this technique? How can we minimize a lot of the work that goes into it? Again, minimize a lot of this labor and time that goes into the actual release glycan method here to get the information that's critical. And the idea was to leverage the PNJS-F enzyme, again, as we release those glycans, and leverage that primary amine that's present on the glycan itself. So how can we leverage some of the standard cross-linking technologies or other click chemistries or anything like that? How can we leverage that to allow for very specific and rapid tagging of that primary amine that's present on the release glycan? This is where the development of the RapiFlora MS reagent came in. So what you'll see here is this is a tagging reagent that leverage, again, kind of standard cross-linking technology you'll see in the green, and that's for very rapid analysis. We have a fluorescence tag, and then one of the more critical pieces of this was the introduction of a tertiary amine for that charge-based detection through mass spectrometry. And this is very important as we try to ionize glycans. Unfortunately, these are not very stable species within a mass spec. And what we can actually do is leverage that tag to localize the charge and minimize a lot of that in-source degradation that you may historically see with glycans. It's a very fast reaction as well. Again, we're knocking down how long this takes. These tagging reactions can operate within seconds. We can leave this and it can actually finish within minutes. And the important part of this is actually it's self-quenching as well. So there's not any extra steps that need to be taken to minimize anything like over tagging or excessive reagents that are present. So again, fast tagging of the actual primary amine is very specific to that primary amine on the glycan. And then again, quenching of the actual tag itself, the excess reagent. So we're not over tagging and causing problems with our separations and detection. So we look at a standard conventional workflow. You're seeing something like greater than 24 hours, moving from a glycoprotein to the actual tagged and cleaned up sample ready to be analyzed. And with this approach for leveraging GlycoWorks, we can actually see we're moving into significantly faster within less than 24 from 24 hours down to potentially just an hour, depending on how many samples have to be analyzed. So very fast process from the initial release to the actual tagging and then cleanup itself, getting to your separations very quickly. It's a very straightforward process. So again, even just leveraging some technologies that have been within our repertoire for a while, specifically RapidJust SF surfactant. This is an MS compatible surfactant. High temperatures, we're able to denature that protein very fast within a couple of minutes, high temperature, cooling that down and allowing for the rapid release, as well as the rapid release and the deglycosylation. So again, we're talking about within a few minutes the ability to do that, and this is very important again for we're really interested in trying to knock this timeline down, but we're not really concerned about anything around the protein. So this is why we're able to leverage a lot of these high temperatures to allow for that rapid unfolding. It's very important to start breaking that structure down as much as possible to allow for the actual access and release of these glycans. And again, we're not worried about the protein, so any sort of modifications that may pop up or any sort of degradation events that may happen because of that high temperature, it's not a concern because we're actually hoping to get rid of that protein anyways. And then after that release, again, moving into the modification through the tag itself, you'll see it's a very stable, highly stable urea linkage. Again, with that fluorophore as well as the MS tag, self-quenching, very fast process. And very, very simple chemistry to kind of understand there. Unfortunately, that we developed this product initially for the focus on kind of standardized MABs, biosimilars, but we know that next generation therapies, some exist as well as and we see that these are becoming more and more prevalent. And so moving from these MABs to kind of these very higher level fusion proteins, and again, this next kind of generation therapeutic modalities, this next set of platforms. And a lot of these present a variety of problems as these fusion proteins are connected through some very complex chemistry, and then the proteins themselves have potentially very complex structures facilitated through things like a variety of intra and intramolecular disulfide bonds that make this process that when initially developed was very simplistic for glycan analysis, but has made it very difficult again through these complex structures, making it difficult to allow for denaturation and PNGSF to access these glycans, release them, and allow for effective tagging. So we're fully characterizing the entire structure. And again, when we're actually performing biosimilar analysis, you want to make sure that you're fully characterizing in its native form any glycans or any other modifications that exist. So if you actually develop an analysis that doesn't take in consideration these very complex, very highly stable structures, you may not actually be doing that. So what we did was provide just a simple modification to the existing protocol, specifically within the denaturation step. So again, a lot of these new fusion proteins that are coming down the pipeline, and some of that do exist that are falling out of patents. They have, again, very complex disulfide bond structures that need to be broken down to allow for full access by PNGSF for release. So really what we're looking at is the ability to do that quickly, as well as some mixed mode separations to take advantage of the sialic acids that are present on a lot of these higher order structures. So again, when we look at the complex nature of these next generation therapeutics, you'll see that really, unfortunately, if you don't break down those disulfide bonds, some of the glycans may not actually may not be accessible by PNGSF. So if we look at the release of these glycans, if we show the mass spectrometry mass spec analysis on the right there, you'll see that there are still some glycans that could be present. Again, you're not fully releasing those. So when you do the tagging exercise, you may not be fully aware of what the full structure is itself. But if you break those disulfide bonds, you're able to knock down and open up that structure a whole lot more. That's when you start to see the ability to fully release all of those glycans, which is illustrated on the mass spectrometry on the right there. We see a fully aglycosylated form, showing that we are releasing and able to observe all glycan structures that are available. So again, this is It's not a difficult modification to the existing workflow. It's very straightforward. We're simply just adding another reduction step within the denaturation step. So again, we're not adding time onto this process. It's simply just the introduction of some sort of reducing reagent to break down those disulfide bonds and allow for that structure to fully open up and make that glycan fully accessible. And then with that, we see the sample prep of this. Waters has a variety of different types of solutions for glycan analysis. So again, even after this process is finished, when you leverage the sample prep kit itself, how do we actually separate it and then actually detect and analyze the samples themselves? There's a plethora of technologies that exist within an organization to kind of give you a streamlined workflow that, again, once you've actually perform the exercise of prepping your samples, you can immediately separate, detect, and quickly analyze all of these. Again, through different column consumables, mass spectrometry platforms, as well as informatics platforms with libraries to streamline this entire process. Then we're going to finish today's presentation and focus on peptide mapping. The idea of focusing on peptide mapping at the end is charge variant analysis and glycan analysis provide very discrete types of data. We're looking at either, again, just a difference in charge or the actual glycan themselves. But when we look at peptide mapping, it can actually be kind of a jack of all trades and provide you all types of information, including glycan and charge variant. So you can actually pull together a lot of the information. It may not provide you the intact analysis, again, because it is peptide mapping. By function of that, it is digesting the protein itself. But you can see a lot, you can acquire a lot of the information in peptide mapping that you have done in glycan and charge variant analysis. So again, when we look at peptide mapping, Waters has a variety of technologies that allow for the separations, specifically in our MaxPeak technology, as well as again, we have that within MaxPeak, but we have, again, also reverse phase for intact as well. But again, focusing on peptide mapping, we have a variety of chemistries that exist for this type of separations, which allows for again, structural characterization to, again, kind of confirm that primary structure. Our technologies are very amenable to MS as well. Again, that's it's very critical to have these stable chemistries that are able to add onto the front end of a mass spec with very little bleeding or anything along those lines that can actually obscure a lot of your mass spectrometry. As well as, again, allowing us to identify a myriad of different types of protein modifications, from glycosylation to any sort of charge variants that you may see, and leveraging this to confirm your identity through a variety of different types of confirmatory signature peptides, as well as determining, again, that lot-to-lot analysis for product integrity. So this is a very important assay, again, from the very beginning for biosimilarity, again, to determine not only the identity, but also the modifications and confirming those that were present within the originator and are present also within your biosimilar. Again, the sample prep workflow for peptide mapping, very simple idea. It can take quite a while, but very straightforward idea, very similar to the actual glycan tagging exercise itself. We're going to go through an unfolding step, again, denaturation, and then we'll have reduction and alkylation. So breaking down those individual disulfide bonds and then capping them to prevent the reintroduction of those disulfide bonds. We'll then go through a desalting step to remove any of the denaturing salts that were leveraged within the first step, which allows for the effective digestion. Because we are breaking down that protein structure, we are also leveraging an enzyme to further digest it, and those remaining denaturing salts may actually break down and remove a lot of the ability for that enzyme to perform its activity. So we do have to remove that through a desalting step, either through some sort of spin cartridge or gravity flow cartridge to allow for that digestion step, and then sample collection. It's a relatively straightforward process, but it can take potentially a day or longer to perform this. Historically, it's been It's been quite a pain, and on each one of these steps has potential to introduce any number of modifications that would not be native to the protein itself, and can cause artifacts that may cause you to think that the product that you made is not similar to the originator itself. So again, it's critical to develop a process that minimizes any of these artifacts throughout each one of these steps to make sure that you're seeing the accurate nature of that protein to show that, yes, you do have something that's similar to the originator itself. So again, why is this important? So when we look at product comparison, so specifically in the top, Remicade, through a variety of injections, we have specific sets of peptides that exist through the digestion process. So we can run those multiple times. We can see that through each one of these injections, that those peptides do remain the same every single time. So what we're hoping to do is in the bottom is when we're actually recreating this protein itself through a biosimilar, we perform this digestion, we should actually be able to map out almost to the exact same degree the actual peptide space that you see in the bottom. So the peptide mapping should match almost to a T what you see at the top. There may be minor fluctuations, and that becomes, and any of those fluctuations have to be evaluated and determined if they affect efficacy. But in general, what you should be able to see with a peptide mapping is that this is going to provide you the exact same set of critical peaks to determine structure through primary sequence, and then any modifications that maybe exist that alter the separation of that peptide, those should also be present within this. So having that and overlaying those, you should see very similar separations. But again, what we noticed at Waters was that this process, unfortunately, causes a lot of artifacts to be present within your peptide mapping. And again, we want those effectively, you want those peptide maps to almost essentially overlay with one another and look almost the exact same, as close as possible. But unfortunately, if we're leveraging the historical process that can take a day, we can introduce a variety of modifications and potentially even over digestion, non-specific digestions, all of that complicate the chromatographic space and cause problems in terms of whether or not we truly do have the same product or not, as well as complicating your data analysis. So what we did was actually reevaluate this from the very beginning. How can we minimize a lot of the work that's going into this process? Not only make it faster, but minimize any of the actual modifications, sample prep modifications that may be popping up because of the process being used. And again, our idea was, we're not reinventing the wheel. We're still moving through the same processes of denaturing, reducing, alkylating, desalting, and digesting. But how can we streamline this process, and where can we improve to make the development of biosimilar a lot simpler? And you'll see, specifically with the desalting, a focus on our SetPak SEC gravity flow columns to allow for simple removal of the desalting, desalting for the removal of the denaturing salts, as well as the introduction of a new enzyme, specifically rapizyme trypsin, for the effective fast digestion with minimal artifacts. So why our focus on trypsin? Again, the idea of gravity flow columns, that's a very straightforward, relatively straightforward product development, making that as simple and painless as possible to allow for the removal of salts. But the big focus here is on the trypsin. So why focusing on trypsin, and why the massive focus on high quality trypsin? It's critical to understand the purity of our recombinant of our trypsin. As recombinant trypsin has no chymotrypsin or other non-specific cleavage events that happens, very high purity, as opposed to animal derived, which can actually have chymotryptic activity, which causes a variety of different digestion events to occur that we may not have wanted in the first place, which complicates our chromatographic space, making the analysis a lot more difficult. What we also looked at was the autolysis resistance. Can we improve this more? And really, this is through creating a very homogeneous set of methylation events that minimizes the ability for trypsin to digest itself. This is going to be critical, again, because each one of those digestion events of the trypsin is going to introduce another peak that not only causes the analyst to have to go in and evaluate to see, okay, is this an actual peptide that is present related to my product? Is it actually a part of my biosimilar, or is it a part of the actual trypsin itself? So it really complicates that space and causes you to dive in deeper to have to do more analysis, which makes things like simplified QC assays very, very difficult. And we really wanted something, again, more on that was thermally stable as well. Again, we're trying to minimize a lot of the time it takes to perform the digestion, because the longer that it takes, the longer those samples sit, the more modifications due to time, due to the presence of oxygen and different environmental, essentially environmental hazards that are related to protein digestion that can cause things like, again, oxidation, deamidation, et cetera, which can introduce artifacts related to charge variants and so on. So we're really wanting to again, how can we speed the process up? How can we minimize any off-target digestions and minimize the actual self-digestion, the autolysis of trypsin, to minimize a lot of the artifacts that exist that cause and complicate a lot of the data analysis. So we look at this. This is where Rapizyme itself comes in, and what we're seeing here is the ability to introduce, again, a very fast, efficient, fast and efficient 30-minute digestion, as opposed to a lot of these standard kind of 3-hour or longer digestions based off of the protein to trypsin ratio. You'll see with the ability to place in very high levels of trypsin, we're able to minimize a lot of the miscleavages, as well as almost get rid of all autolysis. So minimizing a lot of the actual artifacts that exist, as well as making sure that we're getting full digestion, so we're seeing every peptide that's relevant to our protein. And one of the most critical pieces of this, and one of the pieces of data that we really like to present to show why this is so important, specifically the autolysis peaks that were brought up. Again, it's one thing to have a very fast separations. The historical trypsins that are leveraged, you can introduce very high levels of that trypsin, but unfortunately, as you introduce more and more trypsin, you can cause problems, again, such as different autolysis events, that self-digestion of the trypsin as well, which in the bottom chromatogram, you'll see that introduces a variety of peaks that not only complicate your separations, but it causes analysts have to move in and identify each one of those individual peaks. So are they present due to the actual biosimilar itself, or are they actually part of the trypsin? All that has to be further identified and characterized, which complicates the process and makes the determination of biosimilarity even more difficult. So what you'll see with the introduction of Rapizyme is a very flat baseline, no artifacts related to trypsin digestion on itself, and that's going to be critical, again, to having an efficient peptide mapping assay that minimizes, again, all those artifacts that can complicate your analysis. And what you'll see here is, even with the modifications that do exist, we're not foregoing any sort of performance as well. Sometimes there can be handoffs in terms of things like minimizing these off target, minimizing the self digestion here through through these modifications. But what you'll see is we're still with Rapizyme, we're still getting the percent coverage that should be expected when compared to the leading competitors. But that's going to be very important. So we're providing these high level, these high quality enzymes that allow for much more efficient, faster digestions, but we're not removing any of the actual sequence coverage as well. So you're still getting that performance you need there, just minimizing a lot of the difficulties in terms of data analysis and sample prep. But beyond Rapizyme itself, when we look at peptide mapping, there's some critical issues that pop up with the presence of different acidic peptides. So when we look at some of the challenges that pop up, and these are related to things like within charge variants, you can see things like deamidation events that happen through acidic and the presence of more acidic amino acids. Unfortunately, we'll see things like low recovery, poor peak shape, and tailing, and this leads to a variety of issues in terms of robustness of separation, as well as low sensitivity and high variability. So how do we get past this? Historically, what we've leveraged are passivation, as well as the addition of different mobile phase additives, which complicates mobile phase and actually can make it less amenable to mass spectrometry analysis, or the introduction of PEEK, which has a variety of problems as it does it does change its shape at high pressures. And then the introduction of titanium, and titanium itself is still a metal, so we do still see maybe some good separations, but unfortunately it is still a metal, so we may still see again robustness issues as well as sensitivity problems and variability. And all of this, delays and causes problems with transferability of methods, as well as causing problems in terms of investigations and overall issues in terms of, do we actually have the biosimilarity that we say we do when we're developing our products? And that's where MaxPeak technology comes in. And what this is actually an engineered surface that allows for minimizing the actual interaction of these acidic species with the metal surface itself, which gets rid of a lot of those problems that we were talking about before, where we have low recovery, we have high variability, et cetera. So when we look at what MaxPeak is, it is an engineered surface. So what we're saying is this is not a modification to the chemistry itself. We still have the standard particles that are tried and true BEH, CSH, HSS that you would expect. So all of our actual particles are unmodified. But what we have done is actually modified and engineered the surface in purple that you'll see there, actually the internal surface of the column itself, to minimize the interactions with that hardware. So again, you can swap between the MaxPeak and the standard chemistry, and that resin is going to be the exact same. So this is only a modification, again, to the actual metal surface itself. We've engineered that surface to minimize any sort of interactions with it. It's going to be very critical for these metal sensitive compounds that may interact with that. So again, it is present in all of our particle technologies that you may be aware of, the BEH, CSH, and HSS. When we look at how exactly this is going to help with your method development and determining things like biosimilarity, what you'll see is that with conventional technologies, conventional stainless steel columns, you'll see specific peptides which may be prone to deamidation. You may not actually be able to see if that deamidation is present. Unfortunately, it may get tied up within the tailing of that peak itself, and they may broaden out as well, and you're unable to see them. With the introduction of Premier, with that engineered surface and minimizing the interactions, you actually start to see and minimize a lot of that tailing and see the impurities that may exist. Again, so it's getting some of that charge variant information that may be prone to happening through, again, events like deamidation. So we're able actually to see it, characterize it, and potentially quantify it as well. So very important, critical to determining biosimilarity, understanding if these impurities are actually present or not, or if they're an artifact of your analysis. You'll see with this, again, not only are we able to, again, see the actual peaks themselves, we're actually able to increase the actual responses of these peptides. We're actually able to minimize the RSDs too. Of course, as you have a much lower abundant peak, unfortunately, may be prone to high RSDs, but with the ability to actually increase and our recovery, we can see not only with that increased recovery, we see an increase in reproducibility as well, with significantly lower RSDs within this peak detection. And what you may also notice is with some of these peptides that are very prone to different modifications, acidic modifications, but they may be just highly acidic themselves. What you'll see is we may go from the inability to actually detect those particular peptides, which minimizes our sequence coverage, to actually being able to effectively see those peptides and making sure that we're, again, able to confirm the primary sequence, which is a critical piece of that biosimilarity study. Okay. We went through a lot of topics today. And it was probably a rapid fire of a variety of different techniques, starting from, again, charge variant analysis, with giving some basic ideas behind ion exchange, basic theory, and some method development ideas there, for going from glycan analysis, a variety of techniques that Waters has there, as well as peptide mapping. Obviously, these techniques can be kind of convoluted and confusing at times, and there's a lot of solutions that exist. So hopefully we kind of gave you an idea, a very high level idea of how to actually leverage these for biosimilar characterization. And we provided you some insight into this. And if you have any more questions, feel free to ask us or.
Yeah, sure. Thanks, Andrew, so much for the informative presentation. Before we head into our Q&A, I just want to remind everybody to please join us on the 28th of September for our robust multi-level LCMS workflows for biosimilar comparability assessment given by Samantha Ippoliti. And before we go into Q&A, we just want to ask our audience to please participate in a poll question, which is being prompted on your screen now. So would you like to be contacted? Please answer yes or no that you would like to receive more information about Waters products and solutions. So we'll give a few minutes for those to select. Okay. And I think we have some questions coming in, and please feel free. I know we're kind of at time, but you can submit some questions that you have here. We'll do a live Q&A. So one of the first questions that come through is what are Premier columns and what are the advantages? So I know you kind of covered that, Andrew. Would you mind going over that a little bit?
Yeah, so Premier columns themselves, it's the idea is from our end to minimize any of the interactions from some of these metal sensitive compounds. And the idea was, how can we actually do that? And Premier specifically talks about these engineered surfaces that we've introduced on the stainless steel themselves. So again, it's nothing related to the particles themselves. If you're using a BEHC18, a Premier BEHC18 is going to have the exact same resin. But what we have actually done is we've engineered that surface and introduced a modification that minimizes any sort of interaction with that stainless steel. So it's going to be very critical for, again, metal sensitive compounds from glycans like the sialylated species, highly negatively charged, any of those charged species within your peptide mapping, et cetera, anything that may interact with that hardware and cause for these non-ideal separations. That's what Premier technology is actually doing. So it's minimizing those interactions and allowing for much better reproducible separations that you historically may not have seen with not only our technologies but with competitor technologies as well.
Great, thanks, Andrew. Another one, do you have any solutions for O-glycan analysis?
Unfortunately, we do not have existing solutions for O-glycan analysis that is currently being evaluated. We know that's coming up more and more. Again, with the idea around these kind of next generation therapeutics that are coming up, the O-glycosylation has become a much bigger topic. But we are currently evaluating how we can leverage our existing kit for N-glycan analysis. How can we leverage that for O-glycan? So we are doing that. Now, there is still potential that you can leverage peptide mapping for that O-glycan analysis. There's potential to potentially see some of those modifications, but we do not have a kit specific for that yet.
Great. Another one as well. In regards to some of your peptide mapping that you showed, the trypsin, is that in solution or is that immobilized?
That's going to be in solution. So we've actually removed any need to immobilize the trypsin, again, because it's very highly temperature stable to begin with, and it's completely autolysis resistant. So it's a homogeneous methylation process that we have developed. So that minimizes any of the need to do the immobilization. So it actually is much more ideal for kind of this fast type of peptide mapping. We do know that there exists some out there that are immobilized, and again, for those reasons, for stability, as well as the ability to kind of easily remove the trypsin, to remove any of those peptides that may come up from autolysis. So again, our process removes the need for any of that. So it's actually much more ideal for in solution digestion.
Great. Great, thank you. I know we're kind of over time. If there's any other questions, we could take those, but if I don't see any coming in, we can definitely conclude the webinar for today. Okay, well, thank you everybody for joining, and we hope to see you in our future episodes.